Welcome back from the break. We hope you enjoyed a refreshing drink during the break. Moving into our next session, our first presentation in this session is on the development and characterization of generic drug products containing nanomaterials. Our first presenter, Dr. Darby Kozak, is a Deputy Division Director in FDA's Office of Generic Drugs, where he leads a group of interdisciplinary scientists on the development of new analytical methods and methodologies to evaluate the equivalence of complex generic drug substances and complex generic parenteral, ophthalmic, otic, and inhalation formulations. Our final presentation in this session is on the considerations for the quality, safety, and efficacy of prophylactic lipid nanoparticle mRNA vaccines. Our presenter in this session, Dr. Keith Peden, is the chief of the Laboratory of DNA Viruses in the Division of Viral Products, Office of Vaccines Research and Review, and the Center for Biologics Evaluation and Research. He is a product reviewer, and his regulatory portfolio encompasses various types of vaccines such as viral vectored vaccines, DNA vaccines, and mRNA vaccines. Please join me in welcoming our first presenter, Dr. Kozak.
It's a real pleasure to have the opportunity to speak to you today about some recent research and notable outcomes with the development and characterization of generic drug products containing nanomaterials. In addition, to speak to you a bit more about how FDA's National Nanotechnology Initiative, as well as FDA's Generic Drug User Fee Amendments Research and Regulatory Program, aims to address the challenges of developing and regulating these complex products. These programs offer funded research projects, enhanced interactions with the agency, such as product development meetings. They also aid in the development of both general guidances, such as the recently finalized guidance on drug products that contain nanomaterials, as well as product-specific guidances that outline specific recommendations on developing therapeutically equivalent products. I'm Darby Kozak, the Deputy Director of the Division of Therapeutic Performance 1 and FDA's Office of Research and Standards, that is in the Office of Generic Drugs. Our office is responsible for overseeing the GDUFA research program, the development of product-specific guidances that outline FDA's current thinking on appropriate approaches to demonstrate generic drug bioequivalence, and coordinate and provide the advice for pre-ANDA product development meetings. Before I begin, I'd like to remind everyone that my talk today reflects my personal thinking and opinions and should not be construed to represent FDA's official views or policies. My talk will cover 4 aspects related to the development of complex generic drug products containing nanomaterials. These include what is a generic drug product and the importance of the physical-chemical characterization to support the equivalence of a generic drug containing nanomaterials to its reference listed drug, which is also known as the brand product. In this section of my talk, I will outline the similarities and differences in the information needed to support the approval of a new drug application versus needed for a generic drug application, and what a therapeutically equivalent generic drug is. Next, I will talk about how the Generic Drug User Fee Amendments, or GDUFA, research program facilitates generic drug development. This includes researching new analytical methods to characterize nanomaterials and recommendations to demonstrate equivalence. I will share these examples of 3 product-specific guidances for products containing nanomaterials, including a parenteral colloidal iron product, a liposomal product, and an ophthalmic emulsion. I will then share a few examples of how FDA's Nanotechnology National Initiative and GDUFA research has helped advance analytical methods for characterizing nanomaterial equivalence. Finally, I'll share a few recent examples of notable generic approvals for drugs containing nanomaterials. Generic drugs are an important part of the American healthcare system as they create competition that can reduce drug prices. There have been a number of studies to show how this reduction in prices has saved the U.S. healthcare system billions of dollars each year, and that these more affordable medications have helped ensure patient access and adherence to a therapy. A key component to a patient's access and use of a prescribed drug product is their ability to afford it. Although this is an older graph, it clearly illustrates that generics generally provide a more affordable alternative to a brand name product. In addition, Generics also help diversify the drug supply chain, which can help reduce potential drug shortages. Although 90% of prescriptions filled in the United States are for a generic, many complex drug products, such as products that contain nanomaterials, still do not have an approved generic available. Complex drug products, as defined by the GDUFA II commitment letter, can be of complex dosage form, such as suspensions, emulsions, implants, and ointments, that often have more than one phase or properties that are affected by the manufacturing process. Complex products can also be of complex routes of administration, such as locally acting, and also can have complex active ingredients, such as heterogeneous polymeric structures or naturally derived mixtures that may be challenging to characterize or compare by common analytical methods. As I'll speak about later in my talk, a key need for the development and approval of these complex products are analytical methods to measure the critical quality attributes of the product. Depending on the product, it may include characterization of the nanoscale material properties, such as the size and structure. In general, there are two regulatory pathways for a drug application. Either the new drug application, or NDA, which is commonly referred to as a brand name drug, is any drug product that may have a new molecular entity, new formulation, new route of administration, or new indication that would require specific information or clinical investigation to demonstrate that it is safe and effective. These are also commonly known as the Phase 1, Phase 2, or Phase 3 clinical studies that are done to show that the product is both safe and effective for its intended use. My talk will focus more on the abbreviated new drug application, or ANDAs, that are associated with generic drugs. Every generic drug relies upon the information from a reference listed drug, or NDA, and must contain information to show that it is therapeutically equivalent to that NDA. An ANDA or generic drug cannot be submitted if studies are necessary to demonstrate that it's safe and effective. So the main difference between a brand and a generic application is the generics submit studies to demonstrate bioequivalence, rather than studies to re-demonstrate the same safety and efficacy. FDA-approved generic drugs are generally designated as therapeutically equivalent to the RLD product. This means that it can be substituted with the RLD and will have the same clinical effect and safety profile when administered to patients under the same conditions specified in the labeling. I'll discuss more in the next slide. A therapeutically equivalent product may have differences in formulation and manufacture, but those differences cannot give rise to differences that would otherwise make the product not therapeutically equivalent. This includes any claims of superiority to the RLD. FDA's approved drug products with therapeutic equivalence evaluation, commonly known as the Orange Book, is a public list of the drug products approved under the FDA's Food and Drug Cosmetic Act, along with therapeutic equivalence evaluations, or TE codes, for those products. To be therapeutically equivalent, a generic product must demonstrate that it is both pharmaceutical equivalent and bioequivalent to the RLD. Pharmaceutical equivalence means that the generic drug must contain the same active ingredient at the same concentration as the RLD. It also must be an identical dosage form and route of administration. For parenteral, ophthalmic, and otic products, a generic product should also have the same inactive ingredients at the same concentration as RLD, but can differ in some excipients as per CFR 314.94, provided additional information is provided that these changes do not affect the product's safety, efficacy, and bioequivalence. It is important to note that FDA may refuse to receive an ANDA for a generic product that differs in formulation and does not include this additional information. A generic also needs to contain information to demonstrate that it is bioequivalent to the RLD product. This includes studies or justification that the rate and extent of absorption of the generic drug does not show a significant difference from the rate and extent of absorption of the listed drug when administered at the same molar dose of therapeutic ingredient under similar experimental conditions in either a single or multiple study. As I'll highlight later in my talk, for some complex products containing nanomaterials, FDA may recommend a combination of both in vitro and in vivo studies to support bioequivalence, and this depends on the complexity of the drug product. Section 6B of FDA's recently finalized guidance for industry, drug products including biological products that contain nanomaterials, outlines some general considerations for the development of generic drug products containing these materials. In particular, it highlights that sufficient scientific evidence is needed to demonstrate bioequivalence between a proposed generic product and its nanomaterial containing RLD, because any differences in the physical-chemical properties of a nanomaterial may affect the bioequivalence, pharmacology, and toxicology profiles of that product. The guidance includes that conventional pharmacokinetic studies are likely to be sufficient for assessing bioequivalence of systemically acting oral products, but additional in vitro tests to demonstrate equivalent physical-chemical properties would likely be recommended for non-systemically acting products. However, it's important to note that the types of studies recommended will largely depend on the unique properties and complexity of the product. As product development and regulatory assessment depends on the unique properties and complexity of the product, FDA tries to facilitate generic drug development and improve transparency on its current thinking through posting of product-specific guidances. Started in 2007, FDA's product-specific guidances outline the information and types of studies recommended to support the approval of a generic drug product referencing a specific RLD. Product-specific guidances are FDA's recommendations, and an ANDA applicant can propose an alternative approach that deviates from the FDA-posted guidance, but they should include justification for the alternative approach, including data and appropriate references. We also highly recommend that for complex products, ANDA applicants discuss their proposed alternative approach through PSG via pre-ANDA meetings prior to conducting and submitting an ANDA. This way, FDA can get greater understanding of the proposal and provide feedback as needed. PSGs are currently posted on a quarterly basis, and as of October 2022, there are over 2,032 posted product-specific guidances. Of those, there are 23 PSGs for a complex ophthalmic or injectable product that contains nanomaterial. As part of FDA's recently enacted GDUFA 3 commitment, FDA will post a product-specific guidance for a complex product approved in a new drug application on or after October 1st, 2022. Our GDUFA 3 goals are to post a PSG for at least 50% of those new products within 2 years of the NDA approval and 75% within 3 years. In addition, it's important to be aware that FDA posts a forecast list on the PSG website of the PSGs it is planning on developing and revising in the coming year. I highly recommend everyone to visit the FDA's PSG website and to look at the relevant product-specific guidances as a general starting point if you're intending to develop a generic drug product. Over the next few slides, I'll provide a few examples of the product-specific guidance recommendations for products that contain nanomaterials. The first example here is for a parental colloidal iron product, ferric oxyhydroxide. The formulation, control, and properties of these iron oxide nanoparticles are complex and often highly dependent on the manufacturing process. In addition, absorption of elemental iron from the nanoparticle formulation is a complex process that is regulated by the body due to potential toxicity of the free iron. The PSG for this product therefore recommends both an in vivo pharmacokinetic study as well as a panel of in vitro tests to support bioequivalence. I want to highlight that a recent revision to these product-specific guidances include 2 options for the analyte to measure in the PK study. As I will discuss in more detail later, the newer recommended option is measurement of the colloidal form of the iron in serum, which found to be feasible measurement from a recent GDUFA-funded research project. In addition, the nanoscale properties of the particle size distribution and particle morphology are part of the recommended in vitro tests. As you can see from the TEM image, the ferric oxyhydroxide particles are generally less than 20 nanometers in size and can take on a more elliptical shape. Selecting appropriate methods to characterize these properties is critical. Given this complexity, it's often recommended that orthogonal methods are used to support the conclusions. An example here would be the use of an ensemble particle sizing method, such as dynamic light scattering, in combination with a single particle analysis of electron microscopy or atomic force microscopy, given the size and optical properties of the colloidal system. The next example is for a liposomal product. Liposomal doxorubicin is probably one of the first and most well-known liposomal products. It is also the first liposomal product that is approved generic. Liposome formulations are often seen as advantageous as they can improve the safety and efficacy profile of the active ingredient. In addition, the properties of the liposomes can be tailored both through composition and manufacturing conditions, which can give rise to passive targeting to the disease location, such as the EPR effect. Given this complexity, the PSG for liposomal doxorubicin and most parental liposomal drug products recommend both an in vivo PK study as well as a panel of in vitro tests to support bioequivalence. Key nanomaterial attribute tests include comparative measurement of the liposome size distribution and state of the encapsulated drug. For doxorubicin, which is actively loaded into the liposome, you can see it precipitates in the liposome as a crystalline rod that gives rise to the characteristic coffee bean shape of the liposome. In addition, other critical properties include measurement of the number of lamella, as this can affect drug release, characterization of the grafted PEG layer on the liposome surface, and the in vitro release testing in multiple conditions to illustrate similar drug release profiles, as the drug release can be affected by the nanostructure of the formed liposomes. I'll present some new, more sensitive analytical methods FDA has been developed to measure both liposomal morphology as well as in vitro drug release. My last example is an ophthalmic emulsion. The product is locally acting, and the product-specific guidance recommends two options: an in vivo comparative clinical endpoint study, or an in vitro only based B approach. An underlying complexity of this drug product and the in vitro bioequivalence approach was in the development and assessment of analytical methods to measure key properties such as the globule size distribution. As you can see from the cryo-TEM images and the dynamic light scattering histogram, there are formulation factors that give rise to a polydispersed globule size distribution and non-Newtonian viscosity profile, which needs to be considered when measuring these attributes. Moreover, there's a need for appropriate methods to compare these complex profiles to support equivalence. Please note that I'm only presenting a very small snapshot of the research FDA has conducted on the characterization and methods to measure the critical quality attributes of emulsion products. These other studies have been published in the scientific literature, presented at public meetings and webinars, and include novel methods and critical understanding to measure drug release, drug distribution, globule size distribution, and viscosity. FDA's GDUFA Science and Research webpage has a full list of the GDUFA-funded research outcomes and references if you're interested in looking at these, as well as the other research examples I'm sharing today. As mentioned earlier, FDA's Generic Drug User Fee Amendments, which was recently reauthorized on October 1, 2022 under GDUFA 3, gives FDA access to dedicated funds to conduct research that facilitate generic drug development and approval. This research program funds both internal and external research projects. FDA's GDUFA Science and Research Program has funded approximately $20 million in research in 2020. These research projects cover 4 broad categories, including developing new analytical methods for characterizing complex products, and in particular interest is the development of new and higher resolution methods to overcome challenges in measuring and comparing the critical quality attributes, such as the nanoscale properties of complex products that can be used to support generic drug equivalence. An example of a recent GDUFA research project is a project conducted by the University of Maryland to develop a new liquid chromatography and inductively coupled plasma mass spectrometry method. This method is able to accurately measure the different forms of iron in the plasma of subjects infused with a colloidal ferric oxyhydroxide drug. This included the iron that's still in colloidal form, labile or free iron, transferrin-bound iron, and protein-bound iron in the plasma. The direct measurement of colloidal iron overcomes limitations of other methods that necessitated the measurement of both total iron and transferrin-bound iron. In addition, the direct measurement, as illustrated in the subsequent pharmacokinetic study, illustrated the feasibility and advantages that a crossover PK study design had in the number of subjects required over the currently recommended parallel design study. Other projects have looked at new tools to characterize nanomaterial structure. Example here is electron microscopy combined with imaging software as a powerful tool for characterizing the size and structure of these nanomaterials. In particular, cryo-TEM and SEM provide a unique ability to visualize soft materials such as liposomes and emulsions in an unperturbed state. FDA's Advanced Characterization Facility has extensive experience in cryo-EM and the image analysis, as illustrated by the images of my talk, as well as the number of publications they have on our site. An example of this is the 2020 paper characterizing the different structures in approved propofol emulsion drug products. All three products contain emulsion droplets, liposomes, and a hybrid liposome emulsion aggregate. The image analysis was able to easily differentiate and count these different particle types in each product formulation. A key aspect here was that the emulsion globule size distribution was similar for all of the products tested, and although formulation 3 exhibited a larger number of liposome particles, the propofol active ingredient was found to be in the emulsion globules and not the liposome particles. Electron microscopy provides a higher resolution analysis of these nanostructure properties that's not possible by other methods. However, like other imaging methods, a key challenge is ensuring sufficient sampling size to provide a statistical comparison. Otherwise, the method's often a qualitative support to other methods. It's important to note that new imaging and artificial intelligence software is beginning to address these gaps, and therefore we're likely to see more imaging analysis in drug product submissions. An example of this is the use of focused ion beam scanning electron microscopy combined with imaging analysis or other chemical-specific analysis such as energy dispersive X-ray spectroscopy. This combination enables cross-sectioning of a solid material via ablation using the focused ion beam and image profiling the structural and chemical-rich regions. In this example, which is the outcomes of a recent GDUFA contracted to GM Solutions, the polymer, active ingredient, and void regions of a PLGA microsphere were characterized. This method has the potential to understand how changes in the manufacturing process may impact the microstructure of the product, which may lead to a potentially inequivalent product performance. An example of a newly developed tool to assess product performance is the electroanalytical in vitro release method created by researchers at U.S. Food and Drug Administration's Nanotechnology Core Facility. We're likely to hear more about the research done by this core facility, but I do want to highlight some of the work that they've done in collaboration with the Office of Generic Drugs on developing new methods to characterize liposomal formulations. This unique method shown here enables the continuous and direct quantification of doxorubicin from liposomal formulations and overcomes limitations and inaccuracies of conventional separation analysis methods. The method uses a highly sensitive electrochemical measurement of the reduction of 2 ketones on deoxorubicin that's free in solution. That is, only doxorubicin that's been released from the liposome can undergo this reduction reaction. This method can provide a more sensitive evaluation of potential formulation and manufacturing differences that may give rise to a non-equivalent product. As part of their method development and testing, the group examined doxorubicin release from 3 approved drug products. In addition to new analytical methods to measure nanomaterials, there's also a need to develop more quantitative approaches for comparing the equivalence of these properties. An example here is the GDUFA research into the use of the Earth Mover Distance approach to compare the relative difference between 2 histograms without a prior assumption of the distribution. EMD overcomes limitations of the D50 and SPAN descriptors for non-monomodal histograms. When combined with population bioequivalence statistical criteria, the EMD method can be used to compare any shape histograms and the relative differences between those 2 histograms. An example is the comparison of the non-monomodal globule size distribution of cyclosporin ophthalmic emulsion when measured using dynamic light scattering. Rather than simply comparing the mean and range of the globule size measured, EMD enables a more comprehensive and representative assessment of the histogram profiles for comparison. The pharmaceutical industry is continually developing new and unique products utilizing nanotechnology, and as such, the generic industry is increasingly interested in developing generic alternatives for these complex products. As outlined earlier in my talk, a key aspect of generic development and approval is that the generic conducts the comparative test to demonstrate that it is equivalent to the reference listed drug. I want to highlight a few recent notable generic approvals that contain nanomaterials. In the last 2 years, there have been 2 more generic approvals for liposomal doxorubicin, bringing the total number of approved generics to 4 for this critical oncology drug. Last year also saw the first approval of liposomal amphotericin B product, which is a critical antifungal drug product with considerable toxicity issues if and when not formulated correctly. In 2021 and 2022 also saw the first approval of 3 ophthalmic emulsion generics. 2 of these were for a diflupronate and 1 for cyclosporin. Lastly, the first generic ferromoxetol product was also approved in 2021. This brings increased options and competition to the treatment of the anemia. All these approvals represent considerable research, characterization, and understanding of these complex products and their unique nanomaterial properties. In summary, I hope that I've highlighted the importance of analytical methods in the development and review of generic drug products containing nanomaterials. That a generic product is therapeutically equivalent and fully substitutable for a brand product. That the generic product approval relies upon the safety and efficacy information of the RLD product, and to ensure this and the therapeutic equivalence, a generic product must demonstrate that it is both pharmaceutically equivalent and bioequivalent to the RLD. This may include comparative in vitro and in vivo testing, depending on the complexity of the product. Also, that FDA's recently finalized guidance for industry, drug products, including biological products that contain nanomaterials, gives an overview of FDA's thinking on developing generics for these products, and that FDA's product-specific guidances outline the specific information and types of studies recommended to develop a generic product to an RLD containing nanomaterial. To facilitate this, FDA supports the development and use of new methods to address knowledge gaps and limitations in the characterization of these complex products. The examples I briefly touched on in my talk are only the tip of the iceberg, and I highly recommend that you visit FDA's GDUFA Science and Research webpage for a more in-depth list of projects, research outcomes, and collaborative research funding opportunities. Lastly, I want to acknowledge that the number of approved generic products containing nanomaterials has steadily increased. In 2021 and 2022, the first generic amphotericin B liposome injection, diflupronate ophthalmic emulsion, ferromoxetol intravenous injection, and cyclosporin ophthalmic emulsion were approved. Approval and marketing of these therapeutically equivalent products is anticipated to reduce the costs for these treatments and create a more diversified supply chain. I'd like to acknowledge a few of the internal and external collaborators who have helped work on projects presented in this talk. Those researchers and leaders in OGD who have helped develop and oversee the GDUFA research program, colleagues in the Office of Pharmaceutical Quality who advise on and perform much of the needed research, colleagues and collaborators from the other FDA offices who contribute to the GDUFA research program, and importantly, the number of external GDUFA-funded research collaborators. Finally, I want to thank you all for your participation in this event and look forward to taking any questions during the panel session.
Good morning. My name is Keith Peden, and I'm Chief of the Laboratory of DNA Viruses in CBER FDA. What I'd like to do today is to present my perspective on the development of messenger RNA vaccines and some CMC issues, with particular emphasis on COVID-19 vaccines. This is the obligatory disclaimer. These are my views and they should not be construed to represent FDA's views or policies. My talk today will discuss, first of all, some origins of the SARS-CoV-2, the developmental history of messenger RNA vaccines, how they are produced, some product and CMC issues, how to determine the potency of the vaccines, some preclinical studies what are required, and how to assess efficacy, what to monitor, and what assays to use. And then I'll discuss whether this is a platform technology, and if so, what would that mean for vaccine development. Finally, I'll mention something about SARS-CoV-2 variants and the evolving nature of coronaviruses. So SARS-CoV-2, the disease was first recognized in December 2019 in Wuhan, China. China notified the WHO of a pneumonia of unknown etiology in January 2020. The virus was identified as a coronavirus in January 2020, and on this slide you can see electron micrograph of the coronavirus showing pointing the arrows, the spikes around the outside of the virus, and these are due to the spike protein, and they give the coronavirus its name, crown-like or corona. And a higher magnification you can see that there is a spike protein cleaved from S1 and S2, a membrane protein M, an envelope protein E, and inside the virion a nucleic acid and single-stranded positive strand RNA. The spike protein is the protein to which most neutralizing antibodies are directed. So the virus was thought to have arisen by zoonotic transmission to humans, with bats maybe the most likely reservoir. It was related to severe acute respiratory syndrome, SARS virus, that arose also in China in 2002 and finally cleared in 2004. Because of its relatedness, this virus has been termed SARS-CoV-2. The disease is called COVID-19 for coronavirus disease 2019. China released the sequence of SARS-CoV-2 in January 2020, and this allowed scientists around the world to start vaccine design. In January 30th, 2020, WHO declared SARS-CoV-2 a public health emergency of international concern. And on March 11th, 2020, the WHO declared COVID-19 a pandemic. So when messenger RNAs were proposed as a vaccine, some of our thoughts were, how could a messenger RNA vaccine be produced in sufficient quantities to provide the number of doses needed in a pandemic? And would this type of vaccine be effective? So I'd just like to show the results of phase 3 trials that were presented to the FDA in December 2020, showing how efficacious they were in fact. This is the study from Pfizer-BioNTech with their vaccine BTN162b, and on the y-axis is the cumulative index of COVID-19, on the x-axis days after dose 1. And as you can see in the red dots, there are many more cases of COVID-19 in the placebo compared with the blue dots, which are the vaccine group. And they calculate this out to be about 95% effective. These results were published in the New England Journal of Medicine in 2020. 2 weeks later, in the same advisory committee, Moderna's vaccine results were presented to the FDA. And as you can see, the placebo on the top line and the vaccine group on the lower line, again, this vaccine is very effective. They calculate approximately 94%. And these results were published in 2021. So what are the advantages of nucleic acids as vaccines? There is no need to obtain or propagate the infectious agent. Some of these can require high containment in BSL-3 and BSL-4 labs. Only require the sequence of the antigen of interest. The production of messenger RNA vaccine starts with the DNA template for the expression of the antigen sequence, which can be readily produced synthetically. As such, we can optimize the sequence for optimal expression. We can incorporate various nucleosides such as N1-methylpseudouridine in place of uridine, and this reduces the immune sensing and enhances translation. We can remove undesired RNA secondary structures and cryptic splice sites. We can design in desirable features such as the proline-proline pre-fusion stabilizing mutations for the SARS-CoV-2 spike protein. And importantly, we can readily adjust the sequence to new variants if they arose. Now, the path to messenger RNA vaccine production is not generally accepted to be so long as 3 decades, but this review by a Belgian group showed that this took about 3 decades to produce RNA vaccines. I would argue that in fact it probably is closer to four decades, as I'll show you in a minute. There's some challenges with messenger RNA vaccines as vaccines or therapeutics. RNA is a large anionic molecule that is subject to cleavage by ubiquitous ribonucleases. It is unstable at high pH. Anionic molecules do not enter cells, and they require the charge to be neutralized. RNA itself is reactogenic through the innate immune system, and thus they require a delivery vehicle that protects the RNA from degradation allows it to efficiently enter cells, shields the RNA from the cell's immune sentinels, and releases the RNA from endosomes to allow translation in the cytoplasm. Lipid nanoparticles have proven effective as delivery vehicles. So I'd just like to go over some of the important milestones, as I see it, for messenger RNA vaccines. Art Krieg and Doug Melton in 1984 provided the mechanism by which messenger RNA can be made. So they showed that enzymatic synthesis of messenger RNA from a DNA template using a DNA-dependent RNA polymerase from a phage can produce active messenger RNA. A few years later, Wolf and colleagues shocked the world by showing that if you inoculate mouse muscle with DNA and in vitro transcribed RNA, proteins were produced in the mouse muscle. Nobody thought that RNA could be expressed in vivo when injected. Martinong et al. A couple of years later gave the first possibility of RNA vaccines when they inoculated messenger RNA in liposomes and obtained a cellular response to the influenza nuclear protein. Semple and colleagues really developed the ionizable cationic lipids and showed that they incorporated into lipid nanoparticles are very effective in introducing RNA in vivo. Carricker and Weissman in the 2000s showed that you can control the reactogenicity of messenger RNA by use of modified nucleosides. And this work, for this work, they were granted the Lasker Prize in 2021. The next advances came in the scaled-up production of messenger RNA and the evolution of lipid nanoparticle technologies. And I say this took about 4 decades to get to where we are now. A typical cellular messenger RNA has a 5' cap, which increases the stability and also expression. There's a 5' UTR untranslated region and a 3' untranslated region, and the gene of interest. And at the end of the RNA is a poly A tail, which increases translation efficiency and stability. So how do we produce the messenger RNA? This is generally considered the drug substance. So the gene of interest is cloned downstream of a T7 promoter, and then the linearized plasmid DNA is transcribed using phage T7 DNA-dependent RNA polymerase to produce the messenger RNA. And this in vitro transcribed messenger RNA is the drug substance. The drug product was generally produced by some method such as outlined here. On the left box at the top you see a lipid stock in ethanol, and this consists of a cationic lipid, polyethylene glycolated lipid, a DSPC, and cholesterol. And on the right box is the RNA stock solution in an aqueous buffer. These are mixed using generally a T-mixer now to produce spontaneously the lipid nanoparticle messenger RNA. The lipids maybe need to be adjusted, and then the concentration of the lipid nanoparticle is adjusted. It's sterile filtered and aseptic filling to produce the messenger RNA lipid nanoparticle drug product. So what are these lipids? The ionizable lipid is the critical one that was developed, as I said, by Semple and colleagues. And the lipids that are used are MC3, SM102, and ALC0315. And these are involved in nucleic acid complexation. At pH 5, lipids are positively charged. At pH 7, the lipids are uncharged. They are less toxic than permanently charged cationic lipids. They're involved in membrane fusion and endosomal release. And recently, Drew Weissman's lab has shown that they, in fact, provide the adjuvant activity to LNPs. The phospholipid, generally DSPC, this increases the transfection efficiency, improves structural integrity and stability, and ensures appropriate encapsulation. The pegylated lipid, DMG PEG 2000, is an example. There are two domains on the pegylated lipid, a hydrophilic PEG and a hydrophobic lipid domain. And this determines the size of the LNPs, which are generally 50 to 110 nanometers. They minimize particle aggregation, and such that the particles are more homogeneous. They prevent binding of plasma proteins, and thus extends the lifetime in blood, and they decrease nonspecific immune responses. Cholesterol is the fourth lipid. This increases particle stability, increases fusogenic properties of the particles, and endosomal release. So the structures of the cationic lipids used in the FDA licensed LNP products: MC3 is used in Onpatro, ALC0315 is used in the BNT 162B, and SM102 in messenger RNA 1273. So what were some of the potential safety issues? These were novel vaccines with novel components. So how was it possible to assess safety? What test methods should be used, and would they reveal potential safety issues in the clinical trials? What animal systems would reveal these potential toxicities? And should the individual LNP components be evaluated separately or as the vaccine? And should messenger RNA be evaluated separately for its expression in vivo? And how should the results of biodistribution retention studies be interpreted? These are all issues that we as regulators had to address. So some of the C issues with drug substance and drug products. First, the drug substance. We need to find out ways how to assess RNA integrity. We need to quantify the proportion of the RNA that is capped, polyadenylated, and full length. We need to identify the messenger RNA. So we can do this by sequencing the DNA template. We could sequence the RNA, or you could use PCR primers specific for that RNA. We need to be able to determine how pure the RNA is. For example, are there smaller products available through premature termination of transcription, double-stranded RNA, residual components of the in vitro transcription reaction? For the drug product, we need to be able to evaluate the lipid nanoparticle structure, particle size, distribution, and charge being two of the characteristics. We need to determine the proportion of messenger RNA encapsulated in those LNPs, identify and quantify the impurities that are present. And of course, we have to determine the stability of the drug substance and drug product. And this is through transportation and storage conditions, because sometimes the RNA is made in one facility and transported to another facility for the drug product production. So how to assess the consistency of manufacture? There are several ways to do this. We could use non-clinical studies to show that they are in fact produce the same effect. We could use clinical studies to see whether they induce the same immune responses. Or we could use analytical comparability using physicochemical methods. Fortunately for the manufacturers, we chose the third one, as the other two were not really practical. Potency determination was a big discussion both for us as regulators and also the WHO. So how to evaluate potency when the vaccine type is new and the correlate of protection against SARS-CoV-2 is not known? And it is not known, therefore, what type of immune response it is necessary to elicit. Potency, of course, depends on the uptake of the LNP messenger RNA by cells in vivo and the expression of the antigen. So potency assays have been generally cell culture based, and the readouts for these assays are ELISA, Western analysis, or flow cytometry. All three have been used. But after demonstrating that the vaccine is immunogenic, animal potency assays were not considered practical, desirable, or even able to be validated. So in vitro assays are what regulators generally use. Dosing is done by the amount of RNA in the vaccine. With the advent of multivalent vaccines, there are some specific issues associated with them. How to ensure that each drug product component is manufactured consistently. There are two general ways to make a multivalent vaccine. One could mix the RNAs to produce a single LNP messenger RNA drug product. Or you can manufacture each drug product separately and then mix those drug products. But determining that each messenger RNA is present and quantifying the amount of messenger RNA is similar with both. How to assess potency for each vaccine component, and also how to assess the stability of each component. These are all important considerations that we had to discuss. In preclinical evaluation, it is necessary to determine that the vaccine is immunogenic in animal models. This is required for phase 1 as it is for all vaccines, but the demonstration of protection in animal models is more of a complicated issue because it depends on what model system we are using. In the case of SARS-CoV-2, Syrian hamsters, non-human primates, and transgenic mice have been used. But at what stage of vaccine development should we ask for protection studies? And this has not been required for phase 1. So clinical assays to evaluate immune responses. Obviously, there's a humoral immunity, and to determine whether humoral immunity has been elicited, binding antibodies can be assessed by ELISAs and other methods. ELISA is the simplest. And neutralizing antibodies can be assessed in neutralization assays. There are two general types of neutralization assays. Live SARS-CoV-2 using a plaque reduction neutralization test. This has been considered the gold standard for neutralization test, or a microneutralization assay. In addition, pseudotype viruses have been used. Replication competent or incompetent vesicular stomatitis virus vectors have been used, and also more commonly is the replication incompetent lentiviruses. And these assays are generally used. Pseudotype assays are more amenable to high throughput and multiple samples that are used in clinical trials. Cellular immunity. The assays are not used to assess efficacy in clinical trials because of their very complicated and again not open to high throughput, but are being used to determine the complete immune responses to the vaccines. And it's likely that cellular immunity plays a role in protection and or clearance of the virus. So I'd just like to say a few words about platform technology. Is a messenger RNA production a platform technology? And if so, what would that mean? So what testing will be required for an mRNA vaccine that expresses a different antigen using the same LNP and manufacturing process? Once the manufacturing process is validated and experience accumulated, could a vaccine for a variant of the same virus be considered as a strain change similar to that done with influenza vaccines? What testing will be required for a vaccine expressing an antigen for a different infectious agent using the same manufacturing process? What non-clinical studies will be required and which could be dispensed with based on data from similar products manufactured by the same process? For example, will we need biodistribution studies again, or toxicology studies? So how could a vaccine development be streamlined for new messenger RNA vaccines with different LNPs? So non-clinical testing for messenger RNA vaccines with different LNPs, there are two types. The LNP with the known lipid components, but in different proportions, or lipid nanoparticles with new lipid components for which there is no human experience. In the former case, biodistribution and toxicology studies might be dispensed with. For the second case, biodistribution retention studies will likely be requested because there is no experience with these new lipids. And toxicology studies will also likely be requested. So CMC issues for messenger RNA vaccines with different LNPs. Each product requires its own specifications, as summarized on slide 24 in the presentation. And these, again, summarized drug substance, messenger RNA integrity, percent capping and polyadenylating, identity, and purity. For the drug product, the LNP structure, particle size distribution, charge, proportion of messenger RNA encapsulated, and impurities in the product. There are no settled specifications for messenger RNA vaccines. Each product will be assessed individually. The immunogenicity needs to be determined, the potency assay needs to be developed, and the stability of the drug substance and drug product needs to be determined and monitored over time. I'll say a few words about SARS-CoV-2 and its variants, since that's a very topical issue now. Coronaviruses are RNA viruses, and thus evolution of virus is expected over time. Viruses with increased ability to replicate and to transmit more efficiently in humans will be selected for. Immune escape could be a driver of variant generation. As we have seen over time, experience has demonstrated that immunity wanes, and this is not specific to messenger RNA vaccines for COVID-19. All immunity for all vaccines wanes over time. For the case of SARS-CoV-2, neutralizing antibodies are less effective against some variants, specifically the Omicron variant. So the question arose, should booster doses be given, and if so, which vaccine should be used? Should the vaccines be adjusted to reflect the current circulating strains? These issues were discussed at a recent advisory committee, and again there was no consensus on these points, the last 2 points. So in summary of what I've told you today, I've shown you the results of the phase 3 efficacy trials for the Pfizer-BioNTech and the Moderna vaccines, which allowed the FDA to grant emergency use authorization in December 2020. I've demonstrated how this type of vaccine is produced, some of the manufacturing and product issues with messenger RNA vaccines, how potency of the vaccine is determined, special issues associated with multivalent vaccines, and whether the messenger RNA vaccine manufacture is a platform technology, and discussed what that would mean for new vaccines. Also discussed some of the evolution of SARS-CoV-2 and why vaccines may need to be modified. We just have a few concluding remarks. Messenger RNA vaccines have proven to be surprisingly effective. When we were looking at these vaccines in 2020, nobody thought they would reach 95% efficacious. We were debating whether what we would do with vaccines that were only 40-50% efficacious. Fortunately, we didn't have to make that decision. These vaccines have provided extreme safety. We now have examples in hundreds of millions of doses given in multiple people in many countries. I think people do not realize these vaccines will perhaps be so safe. However, Israeli studies a year or so ago reported a low incidence of myocarditis and pericarditis in some populations. However, it is not known whether this is an issue associated with messenger RNA vaccines in general or specifically with COVID-19 vaccines. This was discussed at an advisory committee this year, and it was generally accepted that we need to find this out because other spike vaccines not made by the messenger RNA platform also have some incidence of myocarditis, so we need to know whether it is the spike that's being expressed from the messenger RNA vaccine or the messenger RNA vaccine itself. And that's important because messenger RNA vaccines are being developed for other infectious disease indications as well as for cancer. And it'd be nice to know if messenger RNA vaccines are inherently at risk of providing myocarditis. It is likely that modifications to the manufacturing process and to LNPs will occur in the future. Thank you for your attention, and I think now we move into a session of question and answer.
Thank you very much to Dr. Kozak and Dr. Peden. Again, we have a number of questions in and we'll capture as many as we can. So I'm going to start with Dr. Kozak because I learn something from him every time and I don't know the question of this answer or the answer to this question. So, can a generic made of nanoparticles with equivalent safety and efficacy, i.e. the test performed in vitro, be accepted by the FDA even if its physicochemical characterization is not completely?
Thanks, Dr. Stevens. This is a good question. It has a couple of components to it. I think it kind of gets back to also the aspect of the risk-based approach of the nanotechnology guidance. So in general, it depends upon what types of product properties are the key components to it that need to be demonstrated to show that the product is therapeutically equivalent. So in this instance here, I think the key question says is that you've got significant information to show that it is considered to be therapeutic equivalent. That is, it has equivalent sort of safety and efficacy, which means that you've got demonstration of pharmaceutical equivalence, and you've also done a bioequivalence study. Now, the types of studies that would need to be done to demonstrate bioequivalence comes back to that key component of that risk. So if this is something like a nanoparticle that is for oral route of administration, systemic absorption, then something like a traditional PK study may be sufficient to demonstrate that it's still therapy equivalent and acceptable as a generic product. But the overall properties, let's say, of the active ingredient, the milled nanoparticles, are different too, or has a physical chemical property difference of those sort of particulates within the sort of oral formulation can be different in that instance. In other instances, I think what you really need to come down to is what are the critical attributes, what are their equivalents, and then using a sort of a basis of information or weight of evidence to explain to the agency why the attributes in the studies that you've done show that the product is therapy equivalent. So therefore we can assume it has the same safety and efficacy profile. And those properties that may not necessarily be equivalent, why those don't affect the overall safety and efficacy profile. So I think that kind of gives a broad sort of interpretation of this question, but hopefully that's helpful to explain that for nanoparticles, it's really the risk-based. Which properties do you think are going to be the key components to demonstrate therapeutic equivalence, and building that sort of story, providing that information, and really providing us the full component of it for the evaluation. Thanks for that question.
Thank you. Dr. Peden, I found a couple of questions that are related. I'm going to read them both to you, and hopefully you can tackle them together. So the first says, in the manufacturing of mRNA vaccines, are the lipids considered excipients or starting materials? Please describe the criteria for this decision. And the second question is, are the lipids considered part of the drug substance, or are they excipients? The latter would require them to be evaluated for ADMET separately from the mRNA. Is that correct?
So, I've lost the questions on my sheet over here, but so whether they're excipients, this was debated, and we decided that they were excipients, and this was to be consistent somewhat with the EMA and WHO, and it seemed that they do fall into the excipient class. Whether the drug substance, can you repeat that because I've lost the sheet on the right about the drug substance version? Let's see that.
Sure. So are the lipids considered part of the drug substance, or are they excipients? The latter would require them to be evaluated for ADMET separately from the mRNA. Is that correct?
In fact, they were evaluated by ADMET, at least the novel ones. For example, the cationic lipids. So that was done. The drug substance is considered by us and WHO and EMA as to be the messenger RNA, because that's the equivalent of the active ingredient. And as an earlier speaker said, excipients do have function, and they're not functionless molecules. But we had decided that the active product, the one that is inducing an immune response, is in fact the messenger RNA, and therefore that is the drug substance rather than, and the lipids are the excipients. I think that covers that.
All right, thank you. Dr. Kozak, you presented some really cool new technologies on slide 17. How can industry access them? Is there a fee-for-service system in?
Thanks, Dr. Stevens. Yeah, thank you. I think that's one of the great components of the research program here at FDA is we do get the opportunity to develop a lot of interesting new technologies to address a lot of challenging aspects with characterizing drug products. That can be both through internal research and as well as external research, kind of shown through my presentation. A key component of also the GDUFA research program is we make all of this publicly available. And so a key component is we always want to make sure that we're presenting it through forums such as this, as well as through peer-reviewed publications, or even through our GDUFA research website where feasible and possible. So what I'd really recommend is that anytime you need to or are interested in sort of the latest sort of analytical methods that FDA may be doing in this, is check our GDUFA research website. On there, you'll see aspects of not only research projects that we're currently funding and undergoing, as well as our research reports, but there's also a really great bibliography there of peer-reviewed publications where that information's been provided with details on how to perform those sort of measurements and other aspects like that. Or in other aspects, if you don't find that there, but you see it's in a bibliography, you can always reach out to the GDUFA research website or through our OGD research email and ask for additional information through that aspect for any of those projects. Thank you.
Great, thanks a lot. Dr. Peden, will the FDA allow for platform data to support the shelf life if the manufacturing process is the same, if the same LNP formulation is used, but if there is a different mRNA, can you bridge that platform data?
Well, that's an interesting question. As everyone knows, these are pretty recent vaccine platforms, and not everything has been worked out on that. I think that's an individual decision. If you want my own view, this will certainly be the case down the road. Whether it's for every single modification, that I think that's up to each of the national regulatory authorities. We have certainly used data for one manufacturer using the same technology with a different antigen to influence our decision on shelf life. But I don't know if I can make a blanket statement on that. I think it will be down the road, but I'm not quite sure that we can make a blanket statement right now. Over.
All right, thank you. Dr. Kozak, can you please explain the RLD for generics, especially whether it is acceptable to use an older generic as an...
Thanks for this question. There's a couple nuances to this question, so I will kind of give a bit of a broader overview of what we mean by reference listed drug or RLD for generic purposes. So the reference listed drug is generally the new drug application that performed the clinical studies or other sort of studies that is the basis of submission for the generic, that the generic is considered therapeutically equivalent to. Now, in the other instances, the RLD is generally also if there's needs to be any sort of comparative studies, some in vivo PK or in vivo clinical endpoint sort of study or in vitro sort of studies that are comparative for a part of the generic drug approval, this is designated the reference standard. Under most instances, the reference listed drug and the reference standard are the same. But when the reference listed drug becomes discontinued, an approved generic that's therapeutic equivalent may be considered the reference standard. So in these instances here, you would actually perform all your comparative studies to that reference standard, but you'd still be considered therapeutic equivalent to that reference listed drug. So that's underneath most instances there where you have potentially an older reference listed drug that may be discontinued and a generic that's come on is now considered the reference standard. The other aspect, which is a more of a niche or a narrow sort of a category of products, especially for old generics, ones that may have been approved before what we call the Hatch-Waxman 1984, or there are sort of aspects there that came in through what we considered an ANDA at that time, but now have been determined to serve as their own reference listed drug. Most of these times in the drug product, you'll see in the Orange Book that the ANDA may actually be designated as both its RLD and RS. But like I said, that's a very small set of drug products where the generic is actually considered the reference listed drug. Thank you for that question.
Great. Dr. Peden, what are your perspectives on the two approaches for multivalent vaccines that you mentioned, whether it was mixing the mRNAs to produce a single LNP mRNA drug product or manufacturing each drug product separately and then mixing the drug?
Yes, that's very interesting. When you make two drug products and then mix them, you have much more control over both the manufacturing and the determining the characteristics of them. And this has certainly been done for, say, if you're mixing a flu vaccine with a COVID-19 vaccine, they often use the drug products and then mix the drug products. You have more control over that. However, I think down the road, I'm not speaking for the sponsors, but I think down the road, what they will do when the manufacturing processes are more understood and we have more familiarity with them, I think they're probably going to mix the RNAs into the lipid nanoparticles together. It requires then fewer steps. But as I said in the presentation, determining the potency of that will become an issue. But I think those kind of issues will be worked out in the future. So for now, I think it's easier to characterize two drug products separately and then you mix them for the vaccine. But I think this is a transitory step that will eventually be worked out for all the potency and stability issues. So I mean, I don't have a perspective that the manufacturers will have on this, but that's just the way I think at the moment it will go.
Okay, thanks. Dr. Kozak, is FIB-SEM the only one that is recommended for porosity measurement for nanoscale drug products? Could BET be acceptable?
Thanks, Dr. Stevens. So FIB-SEM is actually in combination with that AI to kind of look at porosity and drug distribution is a really new technology that FDA is exploring through its GDUFA research program because it's a high resolution, but single particle component or very small sort of subset of overall drug product distribution sort of look. So you're looking at in a high resolution way, the cross-section of a drug product, being able to look internally, externally, be able to see where the locality of each component is, such as these voids with inside the microsphere versus externally in the microsphere. So as part of that, it's an exciting new method that can give you a better high resolution analysis of porosity, but it's not the only way to really measure porosity. There's a lot of other different techniques out there, such as you mentioned the BET method, which is nitrogen absorption, that generally give you on the bulk system a better idea of what the total porosity or surface area of that drug product is. But at certain aspects there, it doesn't really tell you the locality of where those pores may be. Similar, there's also mercury pore symmetry and things like that. I think one of the key aspects here is what's the key property to measure? If the key property is porosity and how important that porosity is to the overall performance of the drug product. So that then comes down to then selectively choosing an appropriate analytical method to capture that property. So in that instance there, you can use a combination of even just a high resolution and sort of a more of an ensemble sort of measurement, or if it's a less sort of important property, the ensemble measurement and a control of that should be sufficient. That's a really good question. Thank you.
Okay, Dr. Peden, the first RNA vaccines had to be stored at sub-zero temperatures, but subsequent batches could be stored under refrigerated conditions. What specific change in the formulation made this possible? I think this is a longer question, so good luck.
Well, the short answer is, if I knew, I wouldn't be allowed to tell you, because that's a proprietary issue. But you will have to ask the sponsors, the manufacturers, that question. I don't think they know at the moment, and it's not true that they can all be maintained at 4 degrees. Certainly one needs to be maintained at -80, and the other one can get by at -20. We do not know the specific reasons for this. I mean, they are working very hard to try to understand what makes them more stable and what makes them less stable, because obviously it's much easier to store a vaccine at 4 degrees than it is at -80, and that's not just for shipping and international use. But I don't think it is known yet. But if I knew what they were, I would tell the manufacturers, but I would not be allowed to discuss that openly in a forum like this. I'm sorry.
Yeah, fair enough. Dr. Kozak, will the new nanomaterial guidance published in April of 2022 apply to micellular formulations that are in the nanometer?
Thanks, Dr. Stevens. Yeah, I think the nanomaterial guidance generally applies to any type of material that has this sort of a two-phase sort of system within the 100 to 1 nanometer sort of range, or any sort of nanomaterial basis. And as explained in that sort of guidance, and as well as through some of our product-specific guidances, it comes down to the overall critical properties of that and its relative risk. So in this instance here, this example of a micellular formulation, it would really depend upon the overall micelle size and how that's formed and the general sort of risk associated with getting that potentially different. So I think this comes down to a key component of, is the micellular formulation a kinetic or thermodynamic sort of system? In the kinetic sort of system, similar to like more of an emulsion sort of thing, where the manufacturing properties can be tuned to give you a specific sort of size range. It's highly important to create that sort of size range, and you get a lot of errors there. That would be potentially where you need a lot more sort of characterization of that overall sort of size distribution and critical quality attributes to ensure that they're similar, especially for an equivalence of a generic product. If it's more of a thermodynamic system, what we have there is where the system basically reaches its lowest energy state when it forms that micelle, that it doesn't really depend upon the manufacturing conditions per se to reach that sort of state. Eventually it will go to that sort of form. In those instances there, what you'll see is probably there may be less characterization or need for critical quality attributes to show that. What you need to simply show is that that's a thermodynamic system that will then run that to that sort of system. So I don't know if that generally answers the overall question, but I think in terms of from the perspective of nanomaterial guidance, I think it's really good to look at it from a risk-based perspective. If you know that it's thermodynamic, it's going to reach that lowest energy sort of state, you simply need to show that, demonstrate this, and that those properties are contained and will always be the same. That means it's a lower risk sort of system, and the characterization that may be done for subsequent control and things like that may not be as high or needed as much as for like a kinetically driven system. Thanks for that question.
Okay, and I think we have time for just one more. We'll give this to Dr. Peden. Can you please help us understand the hazards of vaccine manufacturing with mRNA? For example, modifications that might...
Okay, thanks, Olin. I'm not quite sure what hazards they're referring to. I'd imagine that they're referring to modified nucleosides in the RNA. it's important to note that the modified nucleosides that are in the messenger RNA vaccines used so far are naturally occurring modified nucleosides. They occur in RNAs that are in the cell already. So I don't think there will be additional hazards if similar modifications are incorporated. If they come up with a modified nucleoside that is not naturally occurring, then we will have to discuss that, the regulatory authorities, because, but as yet, the ones that have been used so far are naturally occurring. So therefore, we did not think that there's a specific problem with respect to safety when you're incorporating a naturally occurring nucleoside. I hope that covers that point.
Thank you. I think it did. And I wanted to thank everybody for a great morning session. We're going to take a break for lunch now and reconvene at 12:50. So see you back in about 40 minutes.